Good morning, everybody. It's really my pleasure of being here. Um, it's really exciting to see all of our colleagues and people who work on the clinical side and also the families. And for those of you who represent the families, remember you're not alone. We're trying as hard as we can to understand this disorder. And I'm not going to do much of the introduction. I think Harry already covered much of this. I'm just going to emphasize a few points that are going to be important for my presentation, and which is this disorder features two prominent phenotypes, right? One of them is the severe movement dysfunction, and the second is the epilepsy, which is prevalent in many patients. There's quite a bit of the phenotypic heterogeneity in a disorder, so meaning that there's a whole spectrum that spans from the relatively mild late onset to very severe early forms. And it's important also to recognize that there are a number of different mutations that contribute to this disorder. So it's going to be very important to sort of parse out which mutations contribute which forms of the disease and where that happens. GO, what's the GO? GNAO1 encodes for this protein, which is a heterotrimeric G protein, but it's important to know that is one of 16 different forms of the G alpha subunits that's encoded in human genomes. And these G proteins are important transducers of GPCR signals. And these G protein coupled receptors, what's important to know about them, they are fundamental for many, many, many physiological functions. I mean, because of this, it's perhaps not surprising that disease is fairly devastating. But the good news is that those are also the targets for most of the FDA-approved therapeutics. So it's a very good target for possibly coming up with interventional strategies. Just to recap what we know about the potential involvement of the GO in signaling processes downstream from the G protein coupled receptors, you can break down the G protein signaling into the two different components, this inhibitory types of the signals that are provided by the G protein coupled receptors and also the stimulatory. And sometimes those branches are parsed out in the specific regions in the particular different neurons. I'm going to give you an example of this that's happened in the striatal neurons. But together they form a fairly significant network of things that are happening. There's this stimulatory G proteins that feed in adenylyl cyclase that produces a key signal in messenger, cyclic AMP. And inhibitory types of the G proteins provide the opposite influence into the cyclic AMP formation. And this process is also controlled by the RGS proteins that Henrik is going to talk about that speed up the deactivation of the signaling. So what's important to recognize here is that nearly all of the elements in this network contribute to phenotypically similar disorders. The first mutation actually that my group was involved in back in 2013, that's our entry actually in this field, is the mutations in the stimulatory G protein into this network they call GNAL that causes a primary dystonia. Then subsequently mutations were identified in the key enzyme that generates the cyclic AMP. There are also mutations in the RGS deactivation complex that contribute to much of the similar phenotypic manifestations. And also mutations in the G-beta subunits directly contribute to the disorder. And finally, recently, the mutations in the inhibitory G-alpha-I also cause very similar dysfunction. And my group contributed to place in the GO in this network of players that fundamentally shown that it contributes to the dysregulation of a cyclic AMP seconds messenger cascade. One of the other important things to recognize about the GO that it is involved in transducing the signals through a lot of different G protein coupled receptors. So my group profiled more than 100 different G protein coupled receptors in terms of which channels they use to transmit the signal. And we found actually that the GO can transduce a signal from as many as 80% of the G protein coupled receptors. So meaning that a lot of different systems are going to be dysregulated when this G protein is not functioning properly. So back when we were studying G-alpha-OLF and GNAL disorder, we've established the entire pipeline of how we can functionally evaluate the disorder disruptions that are caused by individual mutations in the G-alpha subunits. And we can comprehensively study those dysfunctions by using the cell-based signaling system that, based on breadth, we can monitor the expression of the G proteins, we can monitor the basal conditions, the ability to transduce the signals, and also the dominant negative activity that these G proteins may have. So fast forward a few years, we started applying this to understand the mechanisms of GNAO1 disorder that caused by a variety of different mutations, profiling them. All of this work, by the way, was done in a fantastic collaboration with Dario Escobar here that's contributed with the clinical work to this. So the basic system goes that we use this breadth system that leaves the G-alpha subunit, in this case G-alpha-O, intact, and uses the bioluminescence transfer approach that tags the beta-gamma subunit with Venus and provides the reporter peptide GRK to monitor how this GO involved in transduction of a signal. So this way, when we evaluate all of these different mutations in a GO, we find that invariably lead to the loss of the ability of this G protein to carry on the signal. We call it this loss of function in the signal transduction pathway. We can also modify this system and then introduce the mutated G-alpha-O on top of the functioning copy of the GO. And in some of the cases, with some of the mutations, we see that they interfere with the ability of the normal copy of the GO to transduce a signal, meaning we call this dysfunction as a dominant negative form, and some of the variants cause this dominant negative dysfunction. We also analyze the ability or disability of this mutations to associate with the G beta gamma subunits, which is essential for the initiation of the G protein signals. And again, we find that some of the variants cause the disruptions in the ability to bind the G beta gamma complex. And finally, one of the critical points for the G protein and G protein activation is the ability to be activated by the G protein coupled receptors. We also use a luminescence strategy to study the ability of the mutants to be activated by the dopamine receptor in our model. And again, we find an interest in bidirectional changes in this process, that some of the variants lose the ability to be activated by GPCRs, and others actually stick to the G protein coupled receptors, forming the dominant negative activity. And more recently, we started also analyzing the kinetic aspects of how well the G proteins are, how fast the G proteins get activated by the G protein coupled receptors. And we find examples where the mutations cause a deceleration in ability to be activated, and also look at the converse process, the deactivation that's catalyzed by the RGS proteins. Again, given that the RGS proteins are contributing to similar dysfunction, and for some of the mutations, we also see the deficits in the ability of being deactivated. And typically those variants that show the deficits in RGS-assisted deactivation are located in the switch 2 region of the G-alpha-O, and I've highlighted some of the ones with a significant ability to be deactivated by RGSs on this slide here. And so clearly we run the whole panel of the assays looking at different aspects. So we can generate the severity scores for how badly those disruptions are, basically coming up with a numerical score of what in different domains, and the expression and the trimer formation and ability to transduce the signal, and also the presence of dominant negative activity for different point mutations. Depends on where they're located in the process. And what we find from this analysis is that there are different mechanisms that contribute to this disorder. There are clearly some of the clear loss of function mechanisms that produce the loss in expression of the protein, or deficits in the heterotrimer assembly. But also there are prominent dominant negative mechanisms, such as change in the ability coupled to the G beta gamma subunits, or gain, for example, constitutive interactions with the G protein coupled receptors. And if you put this in the on a plot that measures both the dominant negative activity and also the loss of function, you would find that there is quite a bit of the spectrum that different mutations have, both in terms of the loss of function activity and also the degree of the dominant negative effect that they have. And in collaboration with Dario, we've extended this molecular scoring and severity scaling that we see from the phenotypic observations in the activity assays also to the clinical manifestations. And Dario's group has been collecting and quantifying the degree of the dysfunctions in the clinical manifestations, and such as like presence of the severity of epilepsy, movement disorders, gross developmental language development, and also assign the clinical severity score. What is interesting from those observations, we can start building the correlative models of which types of the molecular dysfunctions may be contributing to the clinical, the severity of the clinical manifestations. And as you can see, overall, the correlation between the experimental scores that are measured through the experimental assays and the clinical observations is actually not bad. And then you can correlate all of these different molecular perturbations to the clinical severity. And through this analysis, we found that the ability of the G-alpha subunit to bind, to form the complexes with beta-gamma subunits correlate the best with the clinical severity. So that's inability to transduce the signal and engage the beta-gamma subunits seems to be one of the biggest driving forces in the disease progression. All right, so from this moment on, I want to talk more about trying to model what we observed phenotypically in the cell-based assay in the mice and generating the mouse models that we could use with a predictive validity to better understand the mechanism of the disease, but also test different interventional strategies. And a lot of our focus will be also in those investigations will be in the striatum, which is a key brain region that is involved in the movement control in general. A lot of those molecular players that have introduced in the beginning, such as type 5 adenyl cyclase, the GNAL, also G beta 1 are very prominently enriched in this brain region, which makes it even more important to examine the contribution of this region in a brain pathology. And a few years ago, we've demonstrated that viral expression of different isoforms of a GO, pathological variants, recapitulate some of the movement disorders in the mouse models. Both the dystonic-like phenotypes, such as clasping, but also the severe movement manifestations, such as ability to learn the movement tasks and also coordinate the balance. So striatum is the relevant region in considering this disorder. And the particular mutation that we've decided to model is G203R, which is one of the most common mutations found clinically. It is located in a switch 2 region, and patients with G203R exhibit both seizures and a severe movement disorder. And no mouse models have been available, and actually Vladimir's group showed that if you just try to model G203R congenitally, it results in the postnatal lethality of the animal. So we clearly needed to be a bit more creative in designing this model, and hence comes our attempt to do the conditional knock-in mouse model with the G203R mutation. So our approach was the following. We've introduced the mutation in exon 6, where it is, G203R, but we also put the cassette in a conditional allele that contains a normal sequence of a GNAO1 encoded by exons 6 and 8 that is flanked by the LOXP sites. So in the absence of the recombinase that's going to produce a normal copy of the GO, because the splicing is going to go to exon 6, and that's going to contain, from the cDNA, that's going to contain intact sequence, and then a Cre recombinase is introduced. This cassette is going to be excised. You're going to reconstitute the normal copy of the gene, but that's going to contain the G203R pathogenic allele. So they give us an ability to go in into the specific brain regions in a neuronal population and ask the question, which of those brain regions contribute to which symptoms as well of a disease. So we started obviously from introducing this mutation in this striatum first, and we used this striatal-specific Cre driver line, RGS9 Cre, cross this conditional mouse models with RGS9 Cre. And we need to run a whole bunch of validation to make sure that this mouse model recapitulates what we designed this to do. So the GO mRNA was not changed, which was a good thing. We know through sequencing that we successfully introduced the mutation in a heterozygous state in this mouse model. We looked at the GO protein expression, and it was unchanged, which is again good. And then we started looking at the network of things that GO is involved in. At cyclase, at C5 type 5, we saw a significant downregulation, which may have been expected, and that would indicate that the cyclic AMP system is perturbed. And also GNL, G-alpha-OLF, that contributes to stimulatory input in the AC5 is also downregulated. Those proteins are known to be in a complex and coordinately regulated. But this was fairly selective changes, because some of the striatal-specific genes, such as DARP32, were unchanged. All right, so what about the manifestations that these mice have? We started with the open field test, where we look at both the overall activity of animals, but also we monitor the stereotypic movements, which is part of a disease manifestation as well. And we find that both the activity levels in the stereotypic movements are significantly increased in this mouse model. It's interesting that the activity increase shows this peculiar pattern. In the very beginning of the test, when the mice are just beginning to be acclimated to the novel environment, the hyperactivity is minimal. And then as the animals go on in this testing, the hyperactivity levels are increasing. We also placed the animals on a rotarod, which is an ability to monitor the gross movements and motor coordination. And we run the rotarod at different speeds, just increasing the challenge in this task. And the conditional knock-in in a striatal-specific fashion shows a significant reduction in the ability of mice to sustain the motor activity in this task. We've also looked at the motor learning, in which case you put the animals on the rotarod, but you at the same speed that gets accelerated, and but you do the trials over time on different days. It's just like learning to ride the bike. Typically, mice see an improvement in ability to master this task, but this conditional knock-in in a stratum of G203R, mice essentially can't really learn to stay on the rotarod as the speed gets accelerated. We've also introduced the gait analysis through the DigiGait system, which is high-speed resolution recording of the mice as they basically get placed on a treadmill, how well they can complete this treadmill task. And we're measuring in this task separately, males and females, how long they can actually perform this task for. And depends on how fast the floor moves in this treadmill. And wild-type animals have no problem completing the task that takes about 20 seconds. Regardless of the speed, they would normally complete them. But G203R knock-in mouse models, even at the lowest speed, start showing quite a bit of a disability in this regard. And the females are more prominent to the dysfunction than males. And clearly, as the speed is accelerated and the task becomes more challenging, most of the animals containing G203R in the striatum fail this task. But for the animals that can complete this task, normally we can do the analysis with a bit more fine-grained resolution and look at how well they work, how they position, they fit, and analyze different aspects of the movements, such as how much they brake, propel, and swing. And again, on a variety of different measures, we find significant deficits. So it's more about the inability of animals to coordinate the movements if they have the disruption in the striatum. We've also looked, because seizures is a prominent feature of the disease, and now we're knocking in this mutation specifically in the striatum. We looked at the contribution of striatum to seizure induction in this model, and we challenged the mice with the GABA-A blocker PTZ in a subthreshold dose, which doesn't really cause much of the seizure phenotype in the control animals. And we do this with a very fine grain analysis. For each of the animal, over period of time, we monitor with a high-speed view recording the seizure manifestations. We get a fairly rich data sets analyzing both males and females. Like I said, in the control animals, the seizures are not that prominent, and a conditional knock-in of G203I in the striatum does not make the situation worse. So essentially, there's no increased seizure susceptibility in this model. We've advanced it further to analysis of the synaptic transmission, which is the ability of the contribution, basically, of a dopaminergic signal transduction cascade that uses uses GO to transduce the incoming inputs. And we use a field recordings, in this case, placed in the stimulating electrode right in the striatum. And here are just examples of the response families in the control animals. We see a significant reduction in the synaptic transmission when G203R is knocked in the striatum, and that's quantification of this phenotype, fairly remarkable loss of the synaptic transmission. All right, so what if we go now outside of the striatum? What if we now leave the striatum intact and knock in this mutation in, say, cortex and hippocampus? And we do this in excitatory forebrain neurons using a CAM kinase Cree driver line. So right away we notice that these animals have a decreased viability. They succumb to spontaneous seizures, and we, over period of time of like 100 to 150 days, we barely have animals left. We see a significant reduction in the body weight, so these animals also show reduced ability to thrive. What about the movement? Again, open field. We see a tremendous hyperactivity that they exhibit. The pattern of this hyperactivity and stereotypy is now quite different. In contrast to the striatal knock-in, we see that these animals initially show massive hyperactivity, but when they acclimate to the environment, these activity levels go down to fairly normal levels. And that concerns the stereotypic movements as well. Interestingly, they show no deficits in a motor coordination or motor learning. And this is just an example of the rotor test. They have no problem mastering the fairly challenging accelerating rotor task. Then we evaluated the seizures as, again, the subthreshold injection of PTZ. And here we started seeing quite a remarkable seizure phenotypes. So unlike the striatal specific line in G203R in the cortical and excitatory neurons, we see a significant susceptibility to seizure induction. Most of the animals show high grades on the seizure score. And then we started thinking about treatments. How can we, knowing what we know, come up with different strategies? There's a fundamental report that was done by Simone and Martinelli that in the C. elegans models, caffeine does show the promise. Interestingly, caffeine consumption also improves the movement-related phenotypes in ETC5-related dyskinesias. And based on what we learned from the circuitry in the striatal neurons, we know that the cyclic AMP system is dysregulated in this indirect medium spiny neurons, and the biggest driver of the cyclic AMP formation in this neuronal population are actually adenosine receptors. So the caffeine is a blocker for the adenosine HPA receptors in the striatal neurons, so it would make sense that at least in the striatal G203R model, caffeine might be promising. And this is what we started probing, starting with the open field and looking at the just the hyperactivity first. And we found that a remarkable improvement in that caffeine has on a hyperactivity level. So in case of the controls, caffeine is a stimulant. If you give caffeine to the normal mice, they start running around way more. And that's what you see here by comparing the blue bars. But in G203R that are hyperactive right from the get-go, if you give them caffeine, they actually reduce the level of the activity. We tested them on rotor task as well, and we see some improvement in rotor task, not complete, but some improvement when we give caffeine to the striatal knock-in mice. And analyze the synaptic transmission properties, and we see that caffeine also shows an improvement in the synaptic measures as well. So I've been asked to just summarize everything that I talked about in this one take-home slide, and I think what it shows us is that, especially considering this mouse model, is that this disorder has a rather divergent molecular mechanisms, and it can recapitulate the contribution of different circuits to different disease manifestations. So, for example, G203I excitatory neurons is involved in hyperactivity, but also contributes to the seizure susceptibility, whereas in the striatum, it contributes to also hyperactivity, but mostly to the motor dysfunction in this case. And caffeine may be a promising therapeutic intervention as well. I just got to thank the people who's actually done this work. Those names are listed. Most of what I talked about with the disease modeling was done by Gloria Brunori, who's a very talented postdoc in the lab. I'm forever indebted to fantastic collaboration with Daria, and we're working together with Simone and Serena Galosha on, taking the caffeine further down the road. Incredibly thankful to the support from the families, from the Bau Foundation in particular, and also the Ilya Musayev, who's been invaluable supporter over the years, and that wouldn't be also possible without support from the National Institute of Health. And thank you very much for your attention.